So the types of systems I look at are normally questions around how does a protein function, how does it interact with its neighbors. So we have these highly dynamic biological systems which are really structurally complex. This is a membrane with lots of proteins and lipids and small molecules diffusing around. So they're very difficult to study with techniques that don't have high enough resolution. And added to that, they respond to different triggers. So whether that be interactions between partners or small molecules or drugs, or applied force or specific wavelengths of light, or membrane potentials or temperatures, they can respond structurally and act like a nanomachine and a specific function. But observing these changes is difficult because they're so small and highly dynamic, and because these changes are often very rare, stochastic. It's difficult to kind of capture that change in the kinetics between different states, as well as getting the structural information at the same time. So there's normally a bit of a disconnect between the structure and the dynamics. You can get high resolution structure, or you can get some nice dynamics, and then you have to kind of plug the two together. So we use atomic force microscopy, and the nice analogy that I'm going to use here is if you imagine yourself blindfolded. So most high-speed AFMs work in tapping mode to make sure that you don't destroy your sample. And so if you imagine tapping as you walk and navigate a surface, you would notice that the amplitude drops when you get to a change in height, an increase in height. And so you use your internal feedback. So you sense that change in oscillation in your hand, you process that information in your brain, and you decide to make a step up until the amplitude goes back to the original amplitude that you started. But we don't do the stick, we do it with a very sharp and we might be oscillating around 2 to 5 nanometers or even less on a very soft samples. And we do this to build up the image. So this gives us very high single molecule resolution down to sub-nanometer occasionally. It can perform the experiments in liquid. We can control the triggers that a protein might or a biomolecule might respond to. So changing pH, changing the buffers, adding different proteins, applying force, all whilst we're imaging the sample at a controllable temperature. So typically room temperature, but we can increase the temperature and without any tags or labels. The slight weaknesses are that standard AFM is quite slow, so you have to scan the tip all the way across the surface, and that can take minutes. It's slightly invasive. That there's always the question of how are we perturbing the system if we're tapping on it lightly, try and maintain very low forces. It can be difficult for novice users, but there's progress in this, and we are because we're only feeling the surface. So we might have to map back what we see on the surface to what's internally. So the speed is something that we can talk about here, the high-speed AFM. So now if you imagine yourself running towards the step, it's likely that you would trip up, and that would be any one of the limitations in that feedback loop. So whether you are not tapping fast enough to be able to detect that by the time your foot gets to the step, or whether you're not able to process the information quick enough if you do detect it, or whether you're not able to step up fast enough. So to get to the stage of high-speed AFM, every single step in the feedback loop had to be upgraded. So with smaller cantilevers, with higher frequency tapping, so now they're around 600 kilohertz in liquid, better feedback controls and detectors, and better piezo's that can act fast enough without bad resonance frequencies. So if we take a look at this on a log scale here, the log speedo scale. Typical AFM, you kind of watch it line by line. The fast scan AFM, if you push it, can get up to maybe 10 seconds per frame or a second per frame in smaller areas with low pixel sampling. Whereas high-speed AFM, kind of getting up to the second and beyond eventually. And this was really nicely demonstrated by the study from Toshio Ando's lab. And this is, although there are many very nice studies, this is where the myosin 5 or an actin film, where you can see the steps, kind of feet moving in that process. And so we can look at lots of different processes. These left two movies are actually not technically high speed, fast scan, but you can still look at dynamics in a time-lapse way, even if these are 1 image every 5 to 10 seconds. So things like lipid behavior. So on this left, we've got separated membrane to a critical, so it's fluctuating. Domains of liquid phase are fluctuating, and this is kind of like a model raft system. We can look at antimicrobial peptides attacking membranes. This is this nice kind of diffusion limited aggregation process where the membrane's being destroyed over time. Proteins binding to membranes is MIN-D, MIN-E, binding to membrane parts and unbinding. Diffusion. So this is diffusion at a rate at which we can track the single molecules and start thinking about what are the protein-protein interactions if we're tracking their positions, and we have the resolution to be able to see when two proteins are in contact, all the way down to higher resolution kind of structural dynamics. So looking at protein, seeing domains, thinking about the conformation of that protein, just in response to a trigger. And so one of the nice systems to look at is an Nexin 5. And this is a protein which trimer to these nice triangles binds to lipid membranes containing DOPS. There's a mistake there. There should be DOPS. So negatively charged lipid, and they bind in the presence of calcium. And the thought is that if you have a rupture in the cell membrane, it would lead to an influx of calcium, allowing the annexin to bind to a membrane, and the whole from expanding. This is quite a nice system that we found with when we got Nano. And you can see here that this just after it's been assembled into a 2D lattice, you have this kind of hexagonal lattice of triangles here, and then in the center there's like an opening that allows primer to sit in to the lattice, not strictly part of it. So occasionally in some of these you will a trimer pop in and disappear. If you focus on this kind of central one, we've got 3 holes, and we can track how one trimer, or possibly 2 at sometimes, is moving around between different positions here. So it's